Hey guys, I just wanted to go ahead and look at our first lecture from Gene to Protein, Essential Dogma. Like I said, this lecture was given the other day, and several of you were absent. Okay, so let's go ahead and go through this particular lecture. So first of all, this is our large picture overview of the process of going from gene to protein, or the central dogma. And this is looking at it in a eukaryotic cell. How do we know? Well, we see that there is a nucleus, so that indicates a eukaryotic cell. So the first process that must happen is going to turn on a gene. What does that mean? That means that we are going to convert some information in the form of DNA into the form of messenger RNA. That step is called translation, and that's what this lecture will be focusing on. Then, once that messenger RNA molecule has been created, it moves out into the cytoplasm through a nuclear pore, where it joins up with the ribosome, where translation will occur, and that's going to be the conversion of the messenger RNA message into a functional polypeptide or protein. So that's the steps that we're going to be focusing in on today. Is going to be the first step, synthesis of messenger RNA in the nucleus. Okay, so anyways, let's just talk a little bit about some of the molecules. Our product is going to be a polypeptide, a protein. Remember, proteins are made up of amino acids. Here is a picture of an amino acid, and let's just review really quickly. Remember, all amino acids have a nitrogen, carbon backbone. One end of the amino acid has a carboxyl group, which gives it its acid nature. And the other end has a amino group. That's where the name comes from, amino acid. Okay. The only difference between all 20 amino acids that all life pretty much uses, there are some exceptions. And I do want to also point out there are literally thousands of amino acids, but all life uses only 20 of those amino acids. The only difference between them is this variable R group. And this variable R group is what gives an amino acid its properties. And there are four basic properties. It can be hydrophilic or hydrophobic. Okay. That's just sort of the big breakdowns between those. But how does that happen? Well, the amino acids, or I should say the R group, can be nonpolar. That would make it hydrophobic. Or it could be polar. That would make it hydrophilic. Also, an amino acid can be acidic, or it can be basic depending upon the R group. It can act as an acid, or it can act as a base. So those are my four basic properties: hydrophobic, hydrophilic, acid, or basic. But I also want to emphasize that the acid and basic amino acids are also going to be polar, meaning that they are also hydrophilic. Okay, so in the process of translation, what are we going to do? This is the second step. We're going to bind amino acids together via dehydration synthesis in the ribosome. We're going to bind them together in order to make this polypeptide. I wanted you to notice the purple boxes are all the same components of the amino acid, and above the purple box is the variable R group. And you can see that those are all three of those are different in each one of these cases, and each one of these amino acids also happens to be hydrophilic or polar. All right, just again, some of the differences between RNA and DNA, just reiterating these, making sure we know the differences. Both RNA and DNA are composed of nucleotides. All nucleotides have a nitrogenous base, a 5-carbon sugar, and a phosphate group. Some of the differences between RNA and DNA has the 4 bases: guanine, adenine, cytosine, and thymine. And RNA has guanine, adenine, cytosine, and uracil. So if you ever see something that has no thymine but has uracil in it, you're looking at RNA. And if it has thymine in it, you're looking at DNA. Just some quick questions there. Also, the sugar is different, and that's where the name for these nucleic acids come from. The sugar in RNA is called ribose, and you notice that it has 2 OH groups down here. And deoxyribose in DNA. That deoxy means an oxygen has been taken off, and that oxygen has been taken off right down there at carbon number 2. That's where the deoxy name comes from. Also, there are some differences in prokaryote versus eukaryotes in these two processes of transcription and translation. Since we looked at a eukaryotic cell first, let's look at it down here at the bottom. In a eukaryote, transcription occurs in the nucleus. There's also this process we call RNA processing, which we'll discuss next week. And then the process of translation occurs in the cytoplasm. So transcription and translation take place in different places and at different times in eukaryotic cells. In a prokaryotic cell, that's not the case. Okay, first of all, there's no nucleus, so transcription and translation occur in the same place, and they can also occur simultaneously. Why? Because there's no RNA processing. What's RNA processing? You'll learn about that. Okay, we'll get there. So again, here's just another close-up view of that whole process of protein synthesis. We're looking at a single gene. You remember most mammals have anywhere between 20 and 25,000 genes. Okay, I think we as humans somewhere around 22,000 genes. So in the process of protein synthesis, we're only going to be transcribing and translating one gene at a time. Okay, so we have our DNA template strand up here. Transcription is where we make the messenger RNA based on the DNA template strand, and we have our base pairing rules again, but you can notice here the base pairing rules are somewhat different. A is going to pair with a U, because remember there's no T in RNA. C pairs with a G, just as it does in DNA. A pairs with U once again, because again there's no T. And T would pair with A on RNA. Okay, so just looking at that as our base pairing rules, it takes 3 nucleotides, which is called as 3 RNA nucleotides, to code for a single amino acid. Those 3 nucleotides together are called codons. Okay, and here we have 12 nucleotides, i.e. 4 codons, which code for 4 amino acids: tryptophan, phenylalanine, glycine, and serine. Okay. So once DNA structure was figured out in 1953, okay, people wanted to kind of figure out what does it mean. But even before this happened, okay, even before the structure of DNA was figured out, people were trying to figure out what is a gene. Okay, this word gene came out before the structure of DNA was actually discovered. Because remember, Mendel called them factors. Okay, early geneticists in the early 1900s started calling these Mendel's factors, started calling them genes. They weren't exactly sure what a gene was, but they knew, just like Mendel did, that for most traits there had to have been at least 2 of his factors, or 2 alleles, or 2 genes that code for every trait. One gene from each parent. Okay, 1 allele from each parent. Okay, so what this picture shows is a metabolic pathway. We start with product A and we end up with, we start with reactant A and we end up with product E. These X's are just showing if a defect occurs in any one of these steps, then we're never going to get product E. But this defect can occur in multiple places or just in a single place. All right, so what does this diagram show you? It shows that A is converted into B. How's it getting converted? There's going to be an enzyme that does that. B gets converted to C by yet a different enzyme. C gets converted into D by another enzyme. And D finally gets converted into product E by yet another enzyme. And if there's any sort of mutation in any one of those enzymes, we will not get product E. Okay? So by studying metabolic diseases, such as alkaptonuria, which is a black urine disease, when kids can get this, it's a genetic disorder and it's diagnosed by a baby has this very dark colored urine, okay? Or PKU, phenylketonuria, which is a metabolic defect where we cannot break down phenylalanine. Every single one of us, you included, have been tested for PKU pretty quickly after you were born, like within an hour maybe of when you were born, you were tested for PKU, because if you have it, then we need to know, the doctors need to know so they can prescribe a certain diet regime so you do not build up phenylalanine, which we'll talk about in a little bit here. So here's PKU. It is a autosomal recessive mutation in a gene that codes for phenylalanine hydroxylase. Okay, so this PAH is phenylalanine hydroxylase. What is happening? Well, that gene, phenylalanine hydroxylase, takes phenylalanine, uh, which is an amino acid, and converts it to tyrosine, which is another amino acid. If there's a defect in this, if there's a defect in that phenylalanine hydroxylase, that PAH gene, that's not going to happen. So phenylalanine begins to build up in your system, which leads to, as I say up here, intellectual disabilities, seizures, and mental disorders. Okay, so it's not a good thing to have. And if you do know, if a person is known to have it, they can make sure that they reduce the intake of phenylalanine via a restrictive diet. Phenylalanine is found a lot in meats. If you drink diet soda, if you look at the side of the can, it'll say phenylketonureacs, beware, this contains phenylalanine. It's found in a lot of different sources. So your diet's got to be pretty restrictive in order not to have these issues that can develop. Okay. The idea of a gene, what a gene was, has changed tremendously over time. As I said, by looking at metabolic defects, we started to get some idea that one gene controlled one enzyme. All right, and it was first done by Beadle and Tatum. You don't need to really know that name. In 1941, once again, before the structure of DNA was discovered. The work started in 1941, finished in 1958, so they did benefit. That's a long time, almost 20 years. Okay, they did benefit by knowing the structure of DNA by the time they finished their study. So what did these guys do? They took some bread mold, Neurospora, a little black mold that grows in your bread, hit it up with X-rays to create mutations. And then they took these various mutants and they started to grow them on an agar base, if you will, which is a nutrient that has sugars and carbohydrates and things like that that allow the bacteria to grow. Okay. In most cases, a bacteria, or in this case, bread mold, can grow on what we call a minimal media. Okay. It basically just has sugar in it. And it can take that and it can make all of its own necessary amino acids out of that sugar, that minimal media, so it can grow. We as humans, we can make 11 of the 20 amino acids, okay, that our bodies need. And I think there's 9 essential amino acids that we have to ingest. We have to get those from our diets because our bodies can't make them. It might be the other way around, but I think we have to ingest 9 of them. So anyways, if one of these, what they were doing is when they hit this bacteria or this bread mold up with X-rays, they were creating mutations in the DNA that coded for the enzymes in this metabolic pathway. Okay, so as I say, mutants require different amino acids because it can't make those amino acids, so we have to add them to the site. Each type of mutant lacks a certain amino acid needed to produce a certain enzyme needed to produce a certain amino acid. If there's a broken gene, we have a non-functional enzyme. And so what do they do? Let's look at that diagram here. Here's a picture of it. All right, so here, what we see here, this is the mold that's growing on the top, okay? And we have our minimal medium right here. This has no added amino acids. The second group has the minimal media, plus it has ornithine. The third group has minimal media plus citrulline, and the fourth group has minimal media plus arginine. The wild type, again, that's the one that's normal, not been subjected to X-rays, is able to grow on all 3. It can grow on the minimal media because it can take those carbohydrates and convert it to all of the necessary amino acids it has. Why? Because it has functional enzymes that can do that. All right. So class 1 mutants, this is where the gene mutation is in the first gene of this metabolic pathway. And this is what's the metabolic pathway is we're starting off with a reactant and we have to make arginine. If we can't make arginine, then the bacteria are not going to grow. So if the mutation is in gene A, okay, they're not going to grow in the minimal media because they can't make arginine. But if I take the minimal media and I make ornithine, okay, that's going to allow those mutant bacteria to go through and make citrulline and take citrulline and make arginine so they can grow. If the class 2 mutants, we have that mutation in the enzyme B, which is going to convert ornithine to citrulline, they're not going to be able to grow because they can't make arginine. So they won't grow in the minimal media. They won't grow on the minimal media plus the ornithine because we can't convert ornithine to citrulline. However, if I add citrulline right here, they will now grow on that one, and they will also grow on the other minimal media because we're starting with arginine already. The class 3 mutants, it's in the last enzyme C. And as you notice right here, it's not going to grow in any of these except for the minimal media that already has the arginine added to it. So with this basic experiment, they came up with the idea that one gene equaled one enzyme. However, as you guys know, not all proteins are enzymes. Okay, and other proteins are coded by genes too. So then they came up with the idea of one gene leads to one protein. But as you guys also know, there are 4 levels of protein structure. The primary structure is the sequence of amino acids. Secondary structure is beta helix or alpha helices or beta pleated sheets created by hydrogen bonds between the R groups. The third level structure, tertiary structure, is going to be the properly folded polypeptide, okay, or it might be a properly folded enzyme, and it works. And many proteins, that's all they go to. But as you know, many proteins have this quaternary or fourth level structure, which is 2 or more of these properly folded tertiary proteins coming together to make a functional enzyme. Like hemoglobin. Hemoglobin has 4 chains coming together. So it has 4 different polypeptides that come together to make a functional hemoglobin molecule. Okay, so then they went into this one gene equals one polypeptide idea, but now we know even more, and we now know that defining a gene is actually kind of problematic because we now know that one gene can make multiple polypeptides due to what we'll get into discussing next week is RNA splicing and dicing. And also that many genes code for RNA products such as ribosomal RNA or transfer RNAs or small interfering RNAs or I forget, there's a lot of them. There's one called miRNA, and I forget what exactly it stands for right now. But there's a lot of different types of RNAs that are all coded for by your DNA. So, like I said, defining a gene is problematic. But this is a great little diagram because they are showing us the flow of genetic information within a cell. DNA to RNA to protein is what we call the flow of information. DNA to RNA, of course, is going to be transcription. RNA to protein is going to be translation. Do notice DNA can replicate itself. It is separate from this process. Replication only occurs when a cell is getting ready to divide, i.e. mitosis and meiosis is the only time DNA replication is going to occur. Transcription and translation occur all of the time in a cell. No matter what kind of cell it is, it's happening all the time. Now, the only time transcription and translation may shut down or slow down is actually during the M phase of the cell cycle. When the DNA is in what form, guys, is the DNA during the M phase of the cell cycle? 3 seconds. Hopefully you got it. It is in the chromosome form. Why won't these processes occur when the DNA is in the chromosome form? The genes are inaccessible. That DNA is all punched up really tightly versus during the rest of the cell cycle known as interphase when the DNA is in the chromatin form and all of those genes are going to be accessible. Okay, so once again, from nucleus to cytoplasm, where are the genes? They're on chromosomes in the nucleus. Where are the proteins synthesized? They're made by ribosomes in the cytoplasm. How does the information get from the nucleus to the cytoplasm? That's going to be messenger RNA, and that's what we're going to talk about now, that initial process of creating messenger RNA, i.e. transcription. Okay. Um, remember our base pairing rules, some just information about DNA, okay, and RNA, excuse me, similar to DNA, has a ribose sugar versus deoxyribose, has uracil instead of thymine. It is single-stranded. And the process of transcription is going from DNA to RNA. We call it transcription because if you know what a scribe is, what did scribes do? Well, before the printing press in the 1400s, when that was developed, 1440 by the Gutenberg press, okay, before that was developed, you had scribes that had to hand copy books, generally the Bible from or the Quran or some other books, mostly important books or science books. There were science books before in the 1400s, especially in Islamic societies. Okay, they had to hand copy these books, and they were generally going from the same language, Latin to Latin, or Arabic to Arabic, Old French to Old French, so on and so on. That's copying. And that's what transcription is doing. It's copying the language of DNA into the same language of RNA, which is they're both nucleic acids. They're slightly different, but there's nucleic acid. They're both nucleic acids. Transcription, making copies. Yeah, scribes made mistakes or put their own interpretations in. Well, that's going to happen in transcription too. So it's a pretty apropos analogy. So again, DNA is in the nucleus, and only one strand is going to act as a template strand. Okay, and that's the strand that the enzyme RNA polymerase is going to read. And we have the same problem here as we do in DNA replication, that we can only build the messenger RNA molecule in the 5' to 3' direction. Again, because the nucleotides, the RNA nucleotides, they only have that OH group on the 3' end. So we can only add to the 3' end. So we build it in the we build the molecule in the 5' to 3' end. The enzyme is RNA polymerase. Remember, DNA polymerase adds nucleotides during DNA replication. RNA polymerase is going to add RNA nucleotides to the growing messenger RNA strand. So here's transcription. This is all in your textbook, chapter 16, beginning around page 332. I would highly suggest that you look through these diagrams at your own pace. You understand what is going on in those diagrams because it can get kind of confusing. I would also suggest you might make a chart that separates DNA replication from transcription and translation. When do they occur? Where do they occur? Why do they occur? What's my reactants? What are my products? What are the enzymes? Know that stuff. Create a chart. It would really help you. Okay? So, first thing that has to happen is that a DNA molecule or gene has to be turned on. What does that mean? That means that RNA polymerase has to bind to what we call a promoter region. What's a promoter region? A promoter region is a sequence of nucleotides at the start of a gene. Any RNA polymerase can decode any gene. So how does an RNA polymerase decode a specific gene? Well, it's a lot more complicated than this diagram is showing you. Here we're only showing you RNA polymerase. There are many other small proteins that we call transcription factors that are unique to any gene. And those transcription factors are what is going to specify which genes are being turned on and transcribed into a messenger RNA molecule. Okay? So we start reading at the initiation site, or we're going to start reading at the promoter site, and we're going to read the template strand. Which one is the template strand? It's the strand that goes 3' to 5' in relation to the direction of RNA polymerase. So in this particular case, RNA polymerase is going from left to right. Okay, so the DNA template strand is the bottom one because it goes 3' to 5'. That means that this RNA transcript is going to be 5' here and 3' there. So we're going to build it in the 5' to 3' direction. Now, if the RNA polymerase was coming from the right-hand side, it would be opposite. then it would be reading the template strand would now be the top strand, which is now 3' to 5' in relation to the RNA polymerase direction. So be careful of that on questions. Okay. Then the RNA polymerase is going to move down the DNA, reading the template strand about 20 bases at a time. It's reading it in the 3' to 5' direction and building the RNA in the 5' to 3' direction. Notice down here, there's no proofreading. DNA polymerase, during DNA replication, proofreads and ensures a faithful copy of the DNA. Yes, mistakes are made, but very few. Okay, like 30 for every 3 billion base pairs. RNA polymerase lacks the proofreading ability. Okay, and so it makes an error in about every 100,000 base pairs. So it can make a lot of mistakes. That's okay, because remember, DNA is our $1 million blueprint, and the RNA is your 10-cent copy. Okay, we're not doing the whole genome. We're only doing one gene at a time. It's relatively short-lived. If you make a mistake and you don't make the proper protein, you make another messenger RNA until the proper protein is made. Okay. So here's transcription. This picture is also in your textbook on page 333, I think, or 332. So look at it. Here is my polymerase is moving from left to right. Do notice we're adding new nucleotides to the 3' end where that OH group is. Here's my 5' group. So make sure you look at that in your textbook. Transcription will end when we reach a certain sequence of nucleotides on the DNA, and the polymerase will disassociate, and the messenger RNA transcript is complete at this point, although we will talk next week about RNA processing, so it's kind of not quite complete. So if you want to go ahead and check out this website and look at some of that information, go ahead and click on that link in your slides. You have these slides available to you in Schoology, and you should be able to click on that link and look at a pretty cool video animation. Well, guys, that's it for today, and thanks a lot for watching.